hello all. A nice scientific day that is opening this morning along the lines of the quality presentations that we've had all through the week. So better understanding the trajectories and compensation in the neurodevelopmental disorders. So a whole program with practical applications for all of us, practitioners, parents, and the persons with autism. And a very, very interesting topic. And our very first speaker is Pierre Gressens, who is a neuropediatrician and a researcher at the INSERM unit, which he is the director of. He is, above all of that, director of the GIS that he took over after Catherine Barthelemy. He is an immense researcher. We are really happy to have him here with us. And he's going to talk about the developmental vulnerability and early trajectories. Thank you very much. So, hello all. Hello Nelson, who is sitting just right there. I would like to talk about vulnerabilities and early trajectories, and I added resilience. I won't say much about this, but I think that in the future this is going to be a topic that we will need to look at. So I think this is an atypical presentation of these atypical developments. Do you see the green? The green line at the bottom is the typical development, and the higher you are on the graph, the more deviation from that typical trajectory. And this is influenced by the genetic factor combined with environment. So it's always about the interaction of the two. And under the influence of the management, this trajectory can come back to the green line that is a typical trajectory or stay away. Something that is not much, hasn't been described much. Some children, even if you don't do anything or much, they will go back to the green line, and this is what we call resilience. And this is something we need to understand better, because that might help us redirect. And this will be influenced probably by environmental factors, genetic or family parameters that will lead to this sort of spontaneous. So it's really a question of trajectory, and you can see that it's not the deviation occurs really early. Hence the importance of early detection, because the sooner you intervene, the better. The difficulty is that in France we have put in place some structures that will enable for early detection, but then once you have detected and once the children are known and you understand that there is a trajectory reorientation, the structures available are rare and the social cost is really high. So there is a gap. There is a gap and many loopholes, and you tell the children, the parents, okay, your child is different, but what do we do? All you can tell them is that waiting times are really, really high. So probably that the government today, with the question of deficits that they already have on their hands, will not prioritize this. And of course, and this is very important, as I said, there is a real biological reason, the sooner the better for the intervention. But once you've put a name on something and you suspect that there is an atypical trajectory, you have to be able to take that into account, and this is where we have a problem. As I said, we have genetic factors, and like any anomaly in man, in mammals in general, genetic factors may play a very important role, but it's always an interface between environment and genetic factors. And when we say environment and genetic, you have the term epigenetics that comes. And this can be one of the factors that mediates the interaction. Improvement is not necessarily always through epigenetics, but that can be a mediator. And it is important to understand the epigenetics role. So just a small reminder of biological basics. So in reality, okay, everybody says it's simple, but sometimes you lose people along the way. So just a simple reminder, we all have genes in each cell, that's DNA, and it is kind of frozen. Once you're born, once the fetus is being, designed, the genes remain the same, unless you're in the face of some pathologies such as cancer. The genes will remain the same. If normal, they will continue normal. If abnormal, they will continue abnormal. It's half from the dad, half from the mom, and the genes need proteins. The protein will make the function. And in between the two, nature sort of designed it properly. RNA is a copy of one of the two arms, because DNA is a double helix, double arm. You have two sides of the scale, and RNA is a copy of one of the bars on the side of the ladder. So this is the simple version. You have DNA, RNA, protein, and we've realized that things are more complex than that. For a gene to become an RNA, the DNA must be open. If you were to spread DNA, that is several kilometers in length, but in a cell it's a few centimeters max or millimeters. So the DNA has to fold everything. So, if you have a kind of a ball of wool, it would be very, really difficult to go in the middle. So you have to have a DNA open at that place. If it is open, you can have RNA, and if it's not, you cannot have RNA. And many mechanisms which we call epigenetics, which control the development of the genes, or whether the DNA will open or remain closed. The structure of the gene is there from the start and is not modified unless you have an exceptional circumstance, such as irradiation, for instance. But the opening and closing of the DNA will enable having an RNA or proteins with a very complex mechanism on which environmental factors can have an impact. So we have some work, and autism is one of the NDDs that have been studied. But probably a lot of things apply more or less to other NDDs, such as hyperactivity or other. So more than half the genes that were described as being associated with autism, more than half a code for proteins that are at the level of the synapse. What is the synapse? It's in the nerve endings for information to jump from one neuron to another neuron. This is taking place at the level of the synapse, which is a very small micron level contact area. So in pink, so in the upper left corner you have pink. the neuron that will transmit information to the green neuron. And there is a very narrow space between the two, and all the ovals are proteins. For the information to move from one neuron to another, so all the ovals that are orange, these are proteins that have been demonstrated to be modified in autism. So more than half the genes described in autism are actually genes of the synapse. So autism and other NDDs are actually anomalies of the operation of the synapse, how the neurons speak between themselves. They do not speak the same way in typical persons. That's at DNA level. Just this to remind you of known genes is DNA. Do we have the same thing at the level of RNA or proteins? I said protein is what gives the function or makes the function. It's not easy to study the protein because it's expensive, it takes time. There is about 30,000 proteins in a cell. You cannot study all of them. But for RNA, we can study them for a drastically reduced cost over the past 10 years. It is possible to study them in one go. RNA is one indirect way of studying the proteins, understanding the proteins. This is not 100% true, of course, because in between there are things that happen. When you study RNA, you have a truncated vision of real life, but you can do it at a large scale for not too expensive. So what happens in the brain of an autistic child? Unfortunately, these children died in a road accident. But when you look at what happens on the level of RNA in the brain of these deceased children, when you look at their genes, there is no obvious anomaly. There is no classical gene of autism. But that being said, they have a classical profile of autistic children. When you look at the correlation of with the clinical profile and you take the classical criteria and looking at the RNA that is different between typical and non-typical children at the neuron level. And when you do that, what you see is at the level of the RNA, at the RNA level, you see some neurons are very different and very correlated to the clinical cells. Not all cells in the brain have the difference, present a difference from typical children. Some, only some. And these are the neurons, so-called associative neurons, the ones that are not directly impacted by sight, audition, but they will integrate different informations. It's the integration of the various information which poses a problem. This is where you see the highest differences and correlated with the clinical issues. And when you look at what RNA are modified, different from typical children, you find RNA that will become proteins of the synapse. So whether at the level of genetic anomalies or most of the time will modify the synapse, or anomalies at the level of the expression of genes, and again, you meet the synapse. You go back to the synapse. So somehow these two types of analysis demonstrate clearly that autism and maybe most NDDs are probably anomalies of the operation of the synapse, whether genetically caused or environmentally caused. Just a sidetrack from the synapse question. We have known, and this was an assumption by Jean-Pierre Changeux about 40 years ago, over 40 years ago. But this has been evidenced now experimentally in animal models, more difficult to demonstrate in man. The connection between neurons, if Mother Nature had to understand exactly which neuron was connected with the another one, even Google wouldn't manage, because you would need lots of information. So what nature does is connect in a random way neurons. If they're close to each other, two neurons will make a synapse. And then what happens is a lot of synapses, too many synapses are produced, and then at some point, some, depending on the stimulations, the stimuli, some synapses will be reinforced, and the ones that are not very used, or not very much used, or useless, they are getting pruned. A lot of housekeeping. So you can imagine that everybody is connected to everybody else. So at some point you will cut connections. Hence the importance of the first month of life, because it is very useful in the first 2 years of life to have this pruning phenomenon. And we know experimentally that if we have not enough synapses, things don't work. Communication between neurons does not work, and conversely, if you have too many synapses, things do not work either. And it is important to have the elimination of the useless synapses. If there was a lot of noise around me, you wouldn't hear what I say, and it's important to cut down all of that noise if you want to get the relevant information. So in phase 1, you have the first trimester, the second trimester, and then birth in dotted lines. And the blue line, 2-3 years of life. And this is teenage, the yellow part. And here is us, adult age. You see that synapses are getting produced in utero with a massive production in the first years of life, with this process of stabilization and pruning or elimination, and hence the importance of early detection. When you're here, it doesn't mean that you can't do anything, but you've already passed that important stage. There is a critical stage. So the importance of psychiatric diseases at puberty, because you have a major change at the synaptic level, and then you start losing. I probably must be here, and that's the bad news for today, and some of you must be around here. So we start losing. So, the 1,000 days, the idea of 1,000 days starts at the third trimester up to about 2 years. In terms of brain wiring, it's massive, major, but not everything is lost because you've missed that window. Things do happen at 90 or 95, but much slower and much less efficiently. So I said earlier that in this study of RNA, that is proteins in the brains of typical and divergent brains. So the differences between typical and divergent, if you look more in detail, you see here the higher the bar on this bar chart, L2, L3 are the neurons that associate between the different inputs, but the microglia is a second very important cell. Microglia is a very barbarous name, but to begin with, it was an immune cell. So all organs in your body have an immune cell that will trigger and will go into action when there is an invasion by parasites. They're not very smart, but they will go and fight to eliminate the invader. Front line, they're the first line responders, and they will call other immune cells that are more specialized. The vaccines, for instance, will go and trigger more intelligent cells. But these are the foot soldiers that just shoot. They will go in the field and shoot. And in the brain, we have, in other places, they're called macrophages, but in the brain, they are called microglia. And in blue, the neuron. The red, the astrocytes that are the support cells, and in green, the cells that produce an insulator, electrical insulators, because messages in the brain are actually electricity. But if you have a live wire that is not insulated, then the current electricity would just get lost, and that's the myelin. The yellow is, we used to think just immune cells, but actually no, they are more than that. We have realized about 10 years ago that these cells, they all play a major role during brain development. They're just not just immune cells. In fact, I spoke about the pruning of useless synapses, and these yellow, they have a major role in the elimination of useless synapses. In green, outreach of neurons. It's like a twig. Synapses are not all over the place in the neurons. They are at the level of the spines. In yellow, you have a microglia, and you have a very, very close contact. It's very intimate contact, and a few minutes later, you may see this, and believe me, there is a green in the yellow. The microglia ate up the spine of the synapse. But if you disturb this phenomenon, there is a molecular code. The microglia will eliminate the synapses that say, eat me, the eat me synapses, or eat me, do not eat me. Molecular codes that helps the pruning. In animals, if you disturb this phenomenon, if you have too many synapses, at the adult age, the mice, animal model, the mice will dysfunction. They will have memory issues, cognition issues, broadly speaking. So this pruning phenomena, pruning of synapses by the microglia is paramount. So I spoke a little about genes and also... So what are the environmental factors that can disturb this mechanism? So the evidence level is not the same for all. We have solid elements for some and just assumptions on others. The first is infections, inflammation. So you have an infection and ensue in inflammation. When you look at the known factors that are proven, causes of NDD, and the environmental factors, and namely prematurity. And this is something that has emerged. We used not to think prematurity on autism. We knew that premature babies had a risk of motor issues, but not so much cognitive or social interaction impairments. But prematurity is something major. We may have better figures in France, like 6 to 7%, but worldwide it's 10%. So the thing is, it's not decreasing. At worst, it's increasing. So the impact of prematurity is on cognition. IQ is a very, very bad assessment of cognition, but in practical terms, it gives a tool to compare. So you have here in blue a UK study and in black a German study. And when you have after below 32 weeks of pregnancy, IQ points will be lost along the way. Very premature children will have more risks of memory issues, cognition, etc. But in all NDDs, premature babies will not escape this truth that, life is not just girls do better than boys. And I'm being punished for saying that, losing my slide. And the second thing that we saw is the Scandinavians were able to have almost 100% of the population studied. You know, the guys in the north, when they do a study, they have 100%. We do at best 52%. But they have shown that below 32 weeks, you have 7 times more risk of demonstrating autistic signs. If you are very premature, you will present 10 times more risk of having autistic signs. So 10 times more, that's huge, and it's So the weight of prematurity is as important as the genetic weight, which doesn't mean that premature children do not also have the genetic risk. Sometimes you associate both. So this slide is complicated, but the message is that prematurity is associated with a silent in utero infection. Normally the fetus is protected with barriers such as the placenta, but some bacteria which can be aggressive will find a nuke in the placenta, and they will silently proliferate. The mother does not have an infection sign, but once the bacteria gets out of the placenta, the immune system of the mother will kill it. But in the placenta, the immune system, for a simple reason, is dormant, because the fetus has half the genes of the mom, half the genes of the dad. And if the immune system worked full-blown, there would be no pregnancy ever. So nature is actually okay, because in order for a pregnancy to take place, so the immune system will be inhibited around the placenta and the fetal environment. And then at some point, some local inflammation will be induced that might reach the fetus that has an immature immune system itself. And other reasons for why premature can have inflammations or infections, there can be infections or mechanical ventilation. So premature babies will have successive episodes of inflammation. And we know now that it's not so much the level of inflammation, it's the duration that is important. And it is really correlated with their outcome. Little inflammation, good neurological development, but many infections, too many brushes with infection put at risk of autism. When you look at the brain of a premature child who died, unfortunately, a few days after birth, and when you look at the microglia, there is these immune cells, which are important for the pruning of useless cells. So these are fetuses that were, for non-neurological reasons, there was an interruption of the pregnancy. You have very small, not very numerous ramifications, but in a premature baby of the same age, born in an inflammatory context, you have many more cells that are activated. So they are going to produce toxic factors for the neighboring cells, and you see that brain cells will not develop normally when in contact with those cells. In an animal model of this condition of prematurity in an inflammation context, and you look specifically for the expression of all RNAs in the microglia, each horizontal line is an RNA, and we have about 14,000 lines. In red, overexpressed is the RNA where you have too many proteins for that gene, and in blue, when you have not enough. So what you see, everything in red is actually genes that are involved in the inflammation, and the microglia become super inflamed in the absence of infection. It's not a legitimate or legal inflammation. It shouldn't. And conversely, what's in blue is not expressed enough, and these are proteins that are useful to eliminate useless signals or cells. So these microglia in premature babies will create toxic factors for their neighbors and forget to do their job, which is pruning the useless cells. The higher, the more elimination of useless synapses. So if you stand in the cortex here, you see that in black, the microglia of a normal mouse. It's in red, the inflamed. So it's double penalty for brain in development, proliferation of toxic for everything around, and they forget to do their job, which is pruning, hence all the behavioral problems that we can witness. So something important in development is what we call plasticity. So each brain structure will have a period of maximal plasticity, and which we call the critical periods, a few days, a few months. The neural network is going to be very plastic, and once it has found its pace, the repertory of plasticity will be reduced, and that's the closing of the opportunity window. Plasticity is important for the brain to adapt to adjust to the environment, to learn things, and at some point for it to function properly and not in an anarchic way. That's called anarchy if your brain is just plastic and doing things and changing, and at some point things need to get frozen, and that's the end of the critical learning period. But for the brain to develop, you need to have a large variability and possibilities. So, great opening. The aim of rehab and the therapeutic strategies that we're trying to establish is that if we come after the closing of the a critical period, can we reopen the window of plasticity and make the network plastic again and remodulate to change the trajectory and get it closer to the typical path? We're not there yet. But by doing this, the researchers understanding are getting a better grasp of what is happening. And as I said earlier, most neurons in our brain are excitatory, but for the system to function, so if we just had excitatory, everybody will be shaking all the time. But we have inhibitor neurons, that is, they regulate the capacity of the neurons to regulate. So this is really, inhibitor neurons are really, really important, but they play another function. With inhibition, they will regulate more or less the amplitude or the plasticity of the network. If with little inhibition, the network will be able to do much. That's plasticity. And at some point when nature thinks that it has reached its optimum or maximum, the system will be frozen and the number of possibilities of the network will be limited. And the interneurons of this type, which are very important for the opening and closing of the critical period, we know there is a deficiency at this level. So whatever the image, we know that we have too little, both in the brain of premature children and in the brains of children born at the right term. And you know that part of the brain dysfunction could partly be related to that deficit. It's not a major deficit. You're missing about 10-15%. So this might explain somewhat the tendency to have epileptic crisis in these children. So maybe this is something that we could start looking at, because we have a target, because we know that there is a deficiency. So maybe we can simulate. I was saying a few words earlier about epigenetics, which does not change the gene, but changes its expression, and how the DNA is open or closed. It's a very complicated slide, but just to tell you that there are not many studies, but it's starting on the brains of children killed in road accidents. We think that there might be anomalies of genes, but also anomalies of the epigenetic system that control the development of genes in autism and other NDDs. We used to think that the brain did not speak to anybody else, it was isolated from... No, the brain is in contact with the environment and speaks with other organs, such as the digestive tract. You have as many neurons in your brain as in your digestive tract, we found that. And there is also the microbiota. You have more bacterial DNA in your digestive tract. So it's a third brain or a third compartment, and there is a lot of exchanges. Your intestines and the microbiota speak to the brain. It's not... So it was said years ago by Hippocrates, all diseases start in the gut. And if you look at the diversity of bacteria in the microbiota, if you look at the diversity, it's not the same in children premature compared to the others. And C-section will aggravate things. So, and this is also true in children with autism born at the right term. But the question is, the egg or the hen. We know that the food diet of children of autism is different from other kids. So is their microbiota different from typical children because they eat differently? Or do they eat differently because their microbiota is different? So where do I stand? Still have enough time? No, getting close to the end. Well, no questions. And we know that the other environmental factors, a few words on heavy metals as endocrine disruptors. It's often evoked, but there is not much evidence yet. Mercury, there were examples with massive intoxications, yes, in that case in Japan. But the weak levels that we have, it remains to be confirmed. But here we have more evidence for lead pipes. For lead, yes, there is a connection. It does exist, but lead has so much impact on many mechanisms of development. So if you put lead on any neuron, whatever its stage of development, it will disrupt. And one of the ways it disturbs the brain mechanism is through epigenetics, because lead will really disrupt genetic mechanisms. The opening and closing of the DNA will be impacted by lead, and once it's in there, it will be fixated for quite a long time. This is something that we've known for a long time, because Saturnism, lead-induced problems have been known for quite a while. And depending on where you live, where you grow, where you develop, it might have a neurological impact. And the bisphenol A and B, that is the endocrine disruptors, they are being pointed at in almost all pathologies on the globe. They may have an impact on the brain development. There hasn't been much studies because there's this blanket explanation of, endocrine disruptors. But you have about 1,200 bisphenols, and we just removed not even the tip of the iceberg. We still have all the rest. Drugs, medication, just to give you an example, a typical example is the valproic acid, the anti-epileptic Depakine, which was the most prescribed in pregnant women up to a few years ago. And the number of women that I saw on Depakine from the age of 7, because they had a convulsion at the age of 7, and everybody feared removing that when they were pregnant. So that's one of the health scandals in France. And this is really Depakine-induced NDDs. The good thing is now it's prohibited, but France did take its time banning Depakine. But the mechanism is, again, by modifying the genetic makeup. Experimentally, if you wanted to, valproic acid, and you just disrupt the genetic makeup. And it's one of the mechanisms through which valproic acid induces NDDs. Mistreatment or sensory deprivation. It's not a really nice topic, but sensory deprivation has been studied in the Romanian orphanages during Ceausescu's reign. Children who had no stimulation for months and years, and they were just clothed around and wrapped tightly. But there was no connectivity established. Not having stimuli and social deprivation in the first months or years of life had dramatic consequences on brain operation. And mistreatment so it leads to accumulation. Whatever the type of mistreatment, the more mistreatment in childhood, the higher the risk of psychiatric diseases in adulthood, and it's also a risk of NDD. So, the last point for me, my slides are blocked. Social status, which is a politically incorrect topic. Social status puts a child at risk of NDD. Normally, politically correct would say that, we're all equal, but we are not equal at birth. We have different genetic heritage. We have different socioeconomic conditions. All studies show the same thing. The major risk factor for a negative risk is the socioeconomic environment of the mother. And the rest is nothing besides that. Somehow, socioeconomic environment will put children in inequality. The life milieu of very, very poor people, it's not just the parents' fault, it's the socioeconomic level. And there have been studies on migrant populations, which are really typical examples of, socially impaired populations. So to go back to this first slide, many things happen early in life. This doesn't mean that you have missed the first stop. You should just drop it. And no, the idea is that if the earlier you intervene, the more chances of getting in that window of opportunity. And let me stop here. And thank you.